Hi everyone, and welcome to lab number 1. In this lab we get to explore the scientific method, a little bit of how that process works, as well as how research is based off of that, and then in turn how you're going to be approaching your own labs with the scientific method and a scientific approach. Then the second part of the lab we get to explore some more interactive sides of your lab where you'll be going through some of the movements that the human body can produce, trying to learn the appropriate terms for that, as well as getting to explore on your own bodies or a willing participant certain landmarks or even just identifying muscles in general. I know a lot of you have stated that you're interested in learning that aspect of the human body, so this should be a fun lab for you on that part. So this is more or less covering what I just said, and I'm going to backtrack a bit and make a suggestion that as we go through these labs, definitely read through the lab manual chapter that I've provided to you first. It's going to give you a good idea of background information as far as the concepts we're going to explore, plus the activities that you're going to be responsible for doing, so that you can pay a little bit more attention in these lectures. These mini lab lectures, I guess, this one's going to be a bit longer. They're really meant to be supplementary and help students that prefer getting a little bit of a lecture, but ultimately I think from your lab manuals and the chapters that are put out there for each lab and lab activity, you're going to gain a lot from going through it before going through these lectures. Along that same line, I want to make sure that you guys are also utilizing the lab manual chapters more heavily than the lectures when it comes to exams and quizzes. Now, these lectures are going to be a part of this, but the important thing is to go through your lab manual and ensure you know the information in there. There's going to be a little redundancy. We might cover something a little bit different in a slightly different way in the lecture, but that's just to give you a different view of the same information. It's not meant to be your primary study source. These lectures are again meant to be supplementary and add to your understanding. Okay? So with that, let's move forward and onward into the scientific approach. So the scientific method, what are we going to cover? We're going to figure out what that is more specifically. We're going to figure out how this is going to affect your labs. What's a common research terms and approaches when it comes to exercise physiology specifically. And then last, we're going to cover how the scores you'll be getting when you do your labs, your lab activities. How do people come up with the criteria that your scores are going to fall into? And that should help you deepen your understanding as to what it means if you get a average or excellent or poor, hopefully not, but it happens in these fitness tests and different aspects of the labs you'll be going through. So the scientific method consists of systematic observation, measurement, and experimentation with the formulation, testing, and modification of hypotheses. Now, that's a bit of a mouthful, I know, but the main points I want you to take home from this is the systematic approach and the modification of hypotheses. And that's really important when it comes to science and your approach as you go through your labs in developing a scientific approach to learning and observing the world. So I've just drawn up a mini study here for us to try to wrap our heads around a couple of terms and an approach and a concept to research in your labs. So in this study, you can see I have two groups, starting over here, A and B, and I've started off matching the groups as best I can. So the reason I've done this is because I've come up with this idea. I have this theory that I have a really good running training program. It's 8 weeks. It's bound to make people faster. But how do I prove it? How do I find evidence to support this thought, this notion that I have, this question? Well, I make a study. And so what I match on these two groups, I should really have two different people here at the start line, but I match them on their running speed for 5K. So I know on average both of these groups, when lumped together in a large group of people, they have a similar average running time for 5K. Great, so already I have two fairly similar groups that I start with. I move forward and I have some sort of pre-test measure. Well, maybe I want to double check and make sure things like their training program is similar, their experience is similar, and again, I get them to run a 5K for me, just so I can see, okay, how quick can you do a 5K? Just to double check that I've matched these participants well. And then I introduce my independent variable. With the independent variable, another way of saying that is, that's the thing I've manipulated. That's the part that I've changed between group A and B. The reason I only want to change one thing between these groups is because I can have a very good, I don't want to say assumption, but a very great idea that one thing I changed is what's going to cause an outcome between the two groups, hopefully a difference. This is what I'm hypothesizing. Okay, so this is my running training program, my 8 weeks of super high intense fast training, and this group, all I'm going to do is I'm going to give them a nothing thing that shouldn't really influence them much. So I'm going to give them a little bit of education on maybe running in general and running training, but I'm going to ask them to keep their running training, their programs week to week exactly the same. Okay, so we have this one change, independent variable. Great. Now what do I need to do? I need to measure this. So that's called your dependent variable. I don't change anything with here, I simply measure it. So independent variable, you change it. Dependent variable, you don't. And it's through this systematic approach that we can do something such as see this group get faster, the group A, and group B stay the same, and we can look back because I was very systematic in my measurements and my approach that I clearly, this is the one variable that led to this outcome. And that provides me the support that I need to say, okay, my 8-week training program clearly is the bomb. It like made this group way faster. And that's the value in the scientific method. Now, as we have probably grown to know through our schooling and academic careers, even you as students early on are probably getting a sense that science isn't quite that straightforward. You're absolutely right. So intuitively, most of you can probably imagine some questions already if you look at this critically. Sean, what the heck are you talking about? You matched for running speed and changed a program and your group was faster. Yeah, but you didn't account for things, and the term for this is bias. So confounding variables, things that influence my group that I didn't account for. So bias can come from two places. One can come from myself, the experimenter. You could tell how excited I was that this 8-week program was going to just make people super fast, which I get excited about. So that's my bias. And an example of how this can influence people is a very classic study on experimenter bias, where these researchers had this group of undergrads. By the way, undergrads, you all are the foundation of modern day science, because I think every study for decades now recruit you to do these studies most of the time, and it's great. I mean, if you have a chance to volunteer to do studies, I highly suggest it. But just keep in mind, some of these older studies didn't have the same ethical guidelines, and so it might seem a little deceptive here, but bear with me, okay? Things have changed now. Please don't think this is how science is done today. Okay, so there's these researchers back in 1963, Rosenthal and Forb, and what they did is they had these group of students. They're like, okay, students, we need these rats to run a maze, okay? So they got to run this maze. We need you to train them, Okay, but unfortunately we have this one group of rats that are kind of dull. They take a long time to learn. Sorry, you all randomly get them. And this group, you all are lucky because these are very bright rats. They seem to pick up the maze really quick, this breed. So you're fortunate. Good luck. It's going to be super easy. Go for it. And sure enough, what they found is the dull rats didn't learn the maze as quickly. It took them weeks longer. In the bright group, they learned the maze very quickly. So they picked up this thing like no problem. The crux of it all is the pool of rats were from the same breed, randomly assigned. There was no indication that one group of rats was actually more intelligent or smarter than the other. And it just goes to show that the expectation that the students had put on these rats, and the rats don't know if they're expecting to be bright or dull. They can't hear that communicated to them. But just purely the expectation and the non-measurable influences that the students put on to these rats ended up making the bright group brighter. So keeping that in mind, that's a very purposeful way to influence the results. However, most studies this happens a little bit more subconsciously. So it's good to explore what sort of biases and to try to protect studies from having the experimenter influence the results. Okay. This will come up later in some of the labs. You'll see. So the other part, as I mentioned before, was confounding variables. I probably rattled it off, and everyone was like, "Wait, what? What the heck is that? Can you define that?" I'll do so now. So confounding variables is systematically acting across the study, which means both A and B groups have this influence of whatever this variable happens to be. So in this case, potentially, maybe both A and B groups didn't follow the training program I gave them. Maybe A group did maybe they ended up getting a coach and getting a very tailored program that was like specific to what they needed in order to go faster, and sure enough they became faster. Whereas the B group, they maybe overtrained. They went far beyond anything that the training program had laid out for them. They got injured, they couldn't train as much. Next thing you know, they're done, they're not training at all, and then you have your post-test. So again, it's just any factor that you can think of that would influence the study. When you're going through your labs, you're going to try to implement this idea too. You're going to think, I'm following these procedures, and most of them should be very good if you follow them very closely. But the whole point of the procedures and the way they're laid out is exactly meant to get rid of experimenter and confound biases in what you're doing. So your results should be good. So to kind of wrap up what I've been saying or alluding to as far as how to approach your labs and your lab activities, first off, read the pre-lab in your lab manual chapters that I give you. They'll be released week to week. Then make a guess. So look at the lab activities, make an assumption based off of what you know of how the results of all the different activities within the lab are going to turn out to be. In particular, if you're testing yourself, that'll probably be the easiest. You might have to wait until you find which partner or person in your group you're going to be working with before you can make a guess. But before you actually get them to perform the lab activity, stop for a second, be like, okay, well, I'm looking at this person, they look pretty fit, I'm going to guess that they can do 20 push-ups or a push-up test. And then conduct the experiment. So conduct the experiment again, as I mentioned, as close to the procedures in the manual as possible. And then last but not least, analyze and share and interpret. So you made a hypothesis, gonna get it, third time's a charm, right? That your partner is gonna get 15 plus push-ups on their push-up test. What do you know? They only rattle off 7. That's fine. Why do you think that was? What sort of influences caused that? Did you overassume their capacity? Do you think they were tired? And what do you know about physiology that might have influenced that answer and that outcome? And then you can refine your initial hypothesis. So when you look at exercise physiology, there's a few designs and concepts that really stand out, and is really helpful to understand, to know that when you're conducting your labs, what aspects of research are you employing, and how is that going to affect your outcome? How do you describe what you're working with at that point in time? So if we look at design in exercise physiology research, you're typically looking at one of two things: either field studies or lab studies. Now, obviously, we'll be in the lab most of the time, technically, your lab. And what that'll do is have a certain outcome, because you can control more variables typically. When you're outside in the field, there's more variables, external factors that can influence the results. An example of this would be, let's say, cycling on a bike, or on a stationary bike versus a bike out in the real world. So when you're cycling on a bike, you can think there's a lot of external factors. There's the wind, there's the weather and temperature, there's different obstacles on the road maybe, there's safety as far as vehicles, you have changing in elevation, so the work gets harder and easier as you go along. All of these variables play into your results that you get in a field study. In a lab setting with a stationary bike, you can kind of get rid of all those. There's not really any wind, which would influence your results. There's not any vehicles. There's no changing of elevation when you're on the road. So because of that, we get pros and cons with both. Okay, one has a bit more control of the settings and the surroundings. The other one, so that would be the lab-based one. The field studies, on the other hand, would be more reflective of what a real race or real performance would be. And so that's why you'd want to use those sometimes. Okay, rigor. So now we're getting into a way to describe research. So how rigorous or how good is the research that's being done? And this is any research really. It's kind of fits in the criteria of reliability and validity. So the difference between the two is reliability. When you're talking about something being reliable, it's that across repeated measures, you're getting the same answer for the same thing. So if I was to measure a meter stick with a different piece of, say like a tape measure, it should measure a meter every time. And if that's so, my tape measure is reliable. I'm getting the same answer, 1 meter every time that I measure this meter stick. So that's great. Validity is a little different. It's more of a descriptive term to explain how true is this thing at measuring what it's supposed to. So an example of this would be if I used a long you probably don't remember this, but they used to have bubblegum tape, where it'd like roll up, it was flat, it was just bubblegum. I don't know, it was long, it was about a foot or so. So that'd be like taking bubblegum and being like, yeah, this is a valid way to measure this meter stick. Oh, if I get 2 of these long strings of bubblegum, that's valid. That's gonna give me an accurate measure. When in reality, it's not really. You know, maybe the machines do cut the bubble gum to the right length when they're produced in the factory and sold, but maybe weather conditions causes it to shorten or lengthen, so it's not a very valid way of doing it. Statistics, now we're getting onto the math side. So there's numbers and different ways we can express our research through stats. Now, I know as soon as I say the M word, everyone starts, the heart rates get up and stuff. I was never a huge math person until later into my academic career, and did some physics and the light bulb went on. But with statistics, for this class, you just want to be able to grasp how it influences the results that we get for these standardized norms that we're using, right? So if you've ever done a test and they have criteria like excellent, good, average, poor, less than poor, or terrible, or whatever it happens to be, how do people come up with that? And this is really what this overview of statistics is supposed to help explain. It's a math thing, it's a numbers thing, how they come up with these criteria. And we're going to be using a lot of this in exercise physiology, in particular your labs. So while I want to encourage you in your lab activities to do them, share your results with other classmates, take a look at how everyone does based off of each other comparatively, I also want you to really take a look at the standardized norms, because that's looking at a whole population. And when we do exercise physiology research, you don't even need to necessarily compare across people, say in a classroom. You can really just look at these norms, these studies that have been done with huge numbers of people, and develop this criteria that fits most of the time for most people. And you'll see what I mean by that a little bit more as we dive into this. So to keep things maybe a little bit simpler to understand, I have this graph, okay? And we're going to compare apples and oranges. So say I'm a farmer and I'm interested in figuring out what weighs more. Perhaps it's important for me when I'm shipping or selling things or whatever the case is. And I have two types of trees in the same orchard. I don't think that's good farming practice, but let's just say I do. And I just pick all these different fruits, and they're all in a thing. And then I want to find out, okay, in this random assortment of fruit in my basket, what tends to weigh more often? Is it apples or oranges in this field? So sure enough, because I love graphs as a farmer, I'm going to draw up this normal distribution of frequency. So for every one apple I pull out of my basket, it gets a frequency of one added to the total count. Okay, similar with oranges. Okay, I'm also going to weigh these. So every time I weigh one, it's going to have a certain weight assigned to it. On this graph, I chose grams. I didn't actually weigh apples or oranges, so this could be way off on what the actual weight of an apple or orange is. Don't quote me on this. And then once we compile all that data, we, in this particular case, end up with a graph like this. So where do we start? So for example, my approach is, so as I hit, let's just say I had an apple. I picked one apple out of the basket, and it weighed 6 grams. So what I'm going to do on that is I'm going to add a 1 here. And so what this graph is essentially saying is, out of all the apples I picked, let's say about 20 apples happen to be 6 grams. That's essentially what this is saying. And I do that by looking at my frequency and my grams. Bear with me here, it'll all wrap up nicely. I just want to explain the background of the chart first, and then a new concept, and then how that ties into your norms. So 6 grams, apples here, doof. Okay, and I do the same for 5, 4, 3 to 1 gram apples, and similar over here with 12. And then I do the very same process with oranges. Okay, so let's say 7 gram oranges. There seem to be the most of those. Okay, so that's going to be, let's say, 15, 16. The numbers don't really matter in this, so just to give some solid examples. So here we are, we got 16 oranges. Great, okay. So after I plot them all out, it seems to look like I get these inverted U or bell curves forming, okay? And you might have heard of that. So I have these bell curves, and they kind of have different shapes, and I'm looking at them both. Now comes the important part to ask ourselves some questions, okay? So we got an understanding roughly what the graph is showing us. let's see what's important. What can we actually pull from this that actually matters to us as farmers here, okay? Well, first of all, I can look at the average or the mean, okay? What's the most frequent or common number as far as weight here? And based off of this graph, I can look at this and say, okay, right here at the top of the circle, right here at the top of the circle, or where I got my pointer, sorry, and then where this dotted line goes is where I have the highest frequency of weight of oranges. And so I know most common orange weight is going to be, let's say 7, okay? Now let's look at apples, okay? So apples tend to be a little bit less, let's say 5, but they have a higher frequency, okay? And that's fine. That doesn't matter much because right now I'm interested in what weighs more typically. And then see the median weight, oranges actually on their norm for oranges tend to weigh more, 7 ounces, than apples. Okay, so that's one comparison. Another one I can say is more to do with the distribution. So now we're going to get into a little bit of math here. So on a more kind of intuitive plane here, where this little green line explains something called a standard deviation. Okay. So standard deviation is typically expressed in, say, like a +1 or 2 on either side of this, and we'll explain where I can find about 65% of all the weight of one particular fruit within one standard deviation. And I could draw a line straight down here, and so it's the volume under here. Now, why does that matter? Why do I care about standard deviation, or why do you care about it? Right now, probably not so much. You're not a farmer. You don't care about apples and oranges. But what this tells us is where can you find the most amount of fruit in this criteria? Well, it's within one standard deviation. And the cool thing is once we go to 2 standard deviations, so that's 1, and let's say the exact same distance of that over 1 more, so it's going to end up, say, along here. Again, I'm looking just at apples. That's going to tell me where 95%, so that's including this side and this side of the curve, 2 standard deviations, 95% of all apples are going to have a weight between, let's say, 2 and 10 grams. Okay, so now I know the range and how much is going to happen in there. So let's pause for a second there. Okay, so I know 95% of the time when I pick apples in this orchard, they're going to weigh 2 to 10 grams. So let's connect that to what we're going to be doing in the lab. Okay, you have a standardized norm. You do 10 push-ups. So now on average, I can probably say 10 push-ups for, let's say, males in this case. 10 push-ups, if we're not talking about apples anymore, we're talking about push-ups. Based off of what I know about push-ups and general capacity, that's going to fall somewhere within 1 standard deviation. So if I know that, and I know 65% of people, men, are going to be able to do 10 push-ups, great. I know this, and that's going to be within 1 standard deviation because 65%. To me, that's average. So now I have a criteria based off of how many standard deviations you are away from this population norm. Okay, so hopefully you see how that ties in a bit. And you can see if you do really excellent, you're in the top 5% at least. And so how common is it, let's say, for someone to do 12, or I changed it from grams to push-ups here, sorry. Let's go back to apples. Let's just keep it, backtrack a bit. So I know if I find an apple that's 15 grams, how exceptional is that? Amazing. And then similarly, what if I start growing apples that are under 1 gram? I know those are terrible. Those are not great apples for weight and growth. Maybe I want to check the soil in that area, whatever the case may be. Okay. So again, hopefully you can kind of make that connection between the distribution, distance from the median, and where the criteria might come from on something like this. Okay. All right. So I'm going to try to tie that all together a little bit for you here before we move on to the more movement-based stuff. We'll leave the scientific method for a bit. But being methodical via the scientific method really helps minimize bias. And again, if you take this approach when you're doing your labs, you're going to maximize your learning experience, and you're going to maximize how the quality of your results are going to be more valid. And when you do repeated measures, either between other peers of yours or yourself, if you're doing measures over and over again, you really are going to get more reliable results. Okay, so again, hopefully you see how that the standardized norm, help generate these criteria that we use in exercise physiology, and hopefully that gives you a better understanding of your own results and why they might be where they are, and where to go from there. So as we dive into some structures here, I want to really encourage everyone to do these landmarking on themselves, find people, etc. Okay, so let's pop over to zygote body here. Okay, so we're going to learn about positioning. So if we look at our anatomical positions, what does that mean? Well, when we talk about purely just the anatomical position, that's this: standing, hands out to the side facing forward, feet square, torso square. Okay, I'm not sure what these floating skeleton hands are. You'll just have to ignore those. It's not meant to be there. But that's what we mean by anatomical position. It's a starting orientation for the body. So from here, we can describe how one part of the body is in relation to another as far as its position, okay? So let's skip over and look at proximal distal. So I'm going to pick 2 points. I'm going to pick the shoulder and the hand. And when you think of proximal and distal almost just sounds like distance, okay? So it's going to be the farthest thing away from the midline of anatomical position. So again, this is anatomical position. My midline goes right up and down the middle of my body. And so I'm going to pick this shoulder to hand. So which one's farther away? My hand is, okay? Yeah, I can bring my hand across my body, but you're kind of talking about, from a limb segment, if you're looking at the shoulder all the way down to the hand's going to be the farthest. So to flip that around and use the other term, proximal, if you have a proxy or someone close to you, this is proximal. The shoulder is proximal to the midline of the body relative to the hand. Okay? Let's scoot on back, take a little look. Superior, inferior. So when we have these terms and we're looking at an anatomical position, another way we describe it is higher up and lower down. So if I use the belly button and the top of the head, I'm going to say the head is superior and the belly button is inferior. And again, you can kind of pick a midpoint. I can flip that around somewhere too. I can go lower on the body and say the hip is superior relative to the foot or the knee, which are inferior to the hip. Okay. Next term would be medial lateral. So again, this is in relation to the midline of the body. So if I decide to pick, let's just even use a single limb. Let's use the leg. So I'm going to say the inside or the medial part of the knee is here, and the lateral part of the knee is there. Very similar. Medial sounds like middle, so that should be an easier way to remember it. And lateral, if you know football very well, you have the football, and if you huck it out to the side rather than throwing it, that side throw, or sometimes backwards, but that side throw to the side is lateral. It's called a lateral, right? So you can also think this is the lateral side of the body, okay? So this hip is lateral relative to the belly button. The belly button is medial, okay? Last but not least, posterior, anterior. So that has to do with the forwards and backwards plane, okay, so to speak. So if I look at this body from the lateral view, I would say the posterior side. You can think of your butt as well, your posterior side of your body is here. Okay, so that's your back. This would be a posterior view of the body. I have flipped this guy around. Okay, the front here is the anterior side, or the front. Okay, so anterior, posterior. Anterior view, posterior view. Okay. If I want to take a look a bit more at a specific, let's say, body part, I'm going to go back to the pelvis. So this is the posterior part. I can probably even dial this back a bit. Give me a sec. Here, let's go skeleton view. Perfect. So here's my pelvis. Is this bones? These technically 3 bones kind of put together here. Looks like one giant one. But this pelvis, again, if I look at it from the front and the back, this would be the posterior pelvis, and that would be the anterior pelvis, okay? And you might have heard, if you're talking about posture at some point in time, the anterior and posterior aspects of, posterior tilt of the pelvis. So anterior means your hips tip forward, posterior means your hips tilt back. All right, so body movements. This is the fun stuff. Okay, I'm gonna cut over to some videos now and show you as we go through those. The order is gonna be different than on the slide in your manual, but I just did them in a way that was a bit more intuitive to me. Okay, so yeah, let's just skip on over there. So the first video that I have up is kind of the side profile there, and so you can see me go through the head tilting forward, that's flexion. I'm going to come back to neutral. Head going back is neck extension. Okay, again, if I'm just talking about the neck, that's side flexion or lateral flexion, as you know now, and this is rotation. Okay, lateral rotation out to the side. What else I can show you here is from the shoulder, that's called flexion or forward flexion of the shoulder, and that'd be just right at that shoulder joint. That's extension. Okay, and we'll talk about differences in range of motion later. If I'm starting in anatomical here, still looking at the shoulders, my arms were added to my body, that's called adduction, adduction, and my hands go up out to the side, and that's called abduction. Talking about my shoulders again, that's a shoulder shrug or shoulder elevation. And then when my shoulder blades drop down below the midline there, that was scapular depression. So my shoulder blades right here move down. Now if we're purely talking about the shoulder blades in this one, between the range of the arm here and here, you're going to see the shoulder blades, a little tricky to see here, but they actually pivot and rotate. So that one's better to feel on someone if you can. Okay, so you're going to palpate the shoulder blade. We'll go over how to do that. So that first one where the shoulders jutted forward, that's protraction. And when I pull them back, that's retraction. Okay? Shoulder, protraction, and retraction. And that really helps to identify which part of your body you're speaking about. Okay? So here I'm doing just from the arm, the humerus, I'm rotating medially and laterally. Okay, so that was medial. If I lock down my humerus or my arm there, the upper arm, my wrist here will supinate and pronate. So it's pronate when the back of the hand is showing, supinate when the palm is showing. Okay. Keep shuffling down the body and torso. Here we are now. So these movements are called forward flexion of the spine. You can just see how everything slumps forward. Good. And then I can go into back extension. So I'm just going to lay back. That's extending the spine. This one I'll show a couple of different ways. So here is lateral flexion. So I'm bending off to the side. And the goal with this one is, as you'll see, to move just along the plane. I'm not also tipping forward or rotating. It's purely to the side there. Okay. Moving along, we'll check out hips and feet. Okay. So from here, next up, what we have is from the hip, I'm going to forward kick. So that's called flexion of the hip. Show that one again. That's extension. So the leg comes back. I'll try to show that because it's a small movement when the leg goes directly back. And that's directly forward and backwards. This one you'll see is off to the side. So again, the leg's going away from the midline. It's moving laterally straight out to the side along that plane. That's called abduction, abduction. This one, from out to the middle, is called adduction or adduction. Okay? So it starts out, pulls to the midline. I'm just moving the camera now for a foot view. We'll try to get just from the leg there at the hip. That's internal and external rotation. You also know it now as medial and lateral rotation. Okay, and you can see how that turns the foot purely from the hip. Okay. Taking a look now more down to the foot itself. We're going to have plantar flexion, so just keeping the foot in neutral, that's 90 degrees, and then pointing, plantar flexion, and that's dorsiflexion, right? Pulling back. You can think of a fin on a dolphin or a whale. The dorsifin is the one that sticks up off the back, so you're pulling the ankle back on that one. That's inversion and eversion. So inverted and evert is inverting it out. Okay. And we'll go over the triaxial movements of the ankle there. So there's actually 3 movements that occur. Okay. Good, so continuing forward after the body movements themselves. Again, you might have noticed that flexion came up for the shoulder and the hip. Abduction came up for the shoulder and the hip. Flexion was involved in the arm, the hip, and the spine. You know, so there's definitely repeated versions of how to explain for each limb, and it all has to do with which plane and from what joint that you're moving from. Definitely worth of you to go over again. Definitely worth it doing it yourself. That's going to be a really, really key factor in understanding movements. You know, so go through the list, try to name them all from each joint. In particular, the shoulders, hips, and neck are the main ones. Okay, surface anatomy. So again, we're going to cut over here to zygote body and touch on a few things here. This is just an example slide. These are not the only ones you need to know. If you go to your lab manual, they'll have a whole list that we'll review now for those. Okay, we're going to try a slightly different view here. Hopefully that works for everyone. So here we have our very able and willing model again, 3D to participate. And we're going to start with some bony projections, okay? So again, find these landmarks on yourself. It's going to be much more intuitive this way if you do that. So we're going to start off with the humerus, okay? So the humerus is actually your arm bone. So if you look at it here, that's your humerus. And a way to think of it that might help, and we'll touch on this again a bit, is, you know, if you've ever hit or said that you've hit your funny bone, this is kind of what it's referring to because it's humerus. I know it's terrible anatomy and physiology humor for you there, but I love it, every second of it. It's great. So you'll end up figuring out where your humerus is. You can actually feel it a few places. So if you just squeeze kind of the outside and inside of your elbow, the parts that stick out, those are called the epicondyles. The medial, so the middle, and the lateral epicondyles of your humerus, okay? So that's the arm bone there, okay? The ulna, this is a good one. So again, let's show you what the bones look like underneath. Oh, I just got rid of that. Bring back the bones. All right. So the ulna, I'm gonna zoom in closer for y'all. Okay. So the ulna is going to be this middle bone here. And if we really zoom in on the back of the elbow, which I think is going to be helpful. Whoa, we're getting way up and close. Let's go to this side. Okay, so your ulna actually comes up over top of the back of the elbow bone. So you can probably see that there a little bit better, okay? So it starts medially at your wrist, and you can go all the way up on the inside of the arm, and it actually hooks on the back. So you know the pointiest part of your elbow, it sticks out there? That's that part. That's actually part of your forearm bone. It's not part of your humerus. And that part that sticks out and hooks around here is called the olecranon process, okay? So that hooks around. So you can definitely feel the olecranon on the elbow. That's what you're feeling, all right? And so again, I'm sure most of you are pretty able to guess that. There you go, sticking out at the elbow, the olecranon process of your ulna, okay? Great. Let's scoot on back up to the shoulder, clavicle. So here's your clavicle right along here. It's at the top of the pec muscle and goes to the shoulder, okay? Let's get rid of some of that skin. Oh, I just threw on muscle. Let's take away that skin. Oh, that's scary. Okay, so your clavicle, here you are, runs along the top. You can definitely feel that along. It's called your collarbone. You might know that. It's referred to collarbone as well. And fun fact about the collarbone and the clavicle, is that it is the most commonly broken bone in the adult human body. There you are. So if that's ever happened to you, I'm sure, I always do a little survey, and sure enough, there's someone that has had that for sure, from class, all right? So let's take a little tour de force around to the posterior side of the body. Okay, well, let's zoom out a little bit because I want to see the spine. Okay, we're moving on to the spine. That's a pretty good view right there. What do you think? Okay, so cervical spine, where is that? That's right up here in the top part of the neck. And you can see it has a little bit of an extension curve to it, right? So from here, I don't know if you can really get a good vibe for that, but that's C7. So the 7th cervical vertebrae, C7, sticks out. And you can feel that if you go to the back of your own neck. It is very clearly the first vertebrae you kind of feel. I mean, you can dig into your neck and your cervical spine and feel it, but the first one that juts out right at the top of your shoulders there, that's C7. Along here is your thoracic spine, so the one that curves forward flexion in with your rib cage. That there's 12 vertebrae there. So thoracic spine. You can think of your thorax as your like ribs, or if you know bugs better, their thorax is the main component of their bodies, right? So the thoracic along here, okay? We scoot on down, again there's 12, so let's recap because I want to correct what I said. There's 7 cervical, 12 thoracic, and then we get into our lumbar. This is where there's 5 vertebrae, okay? And again, you notice that it goes back into an extension pattern. So extension, flexion, extension, and that's actually a very ergonomical way to support weight. So you definitely want to keep that with that slight S curve or curvature. Perfectly straight back is actually not correct posture, okay? And then so let's flip around to the front. Next up, we have sternum. Okay, so anterior view, we're looking here, and sure enough, look at this, we have our sternum right here. This is called the xiphoid process. You don't need to know that, but it's the part if you reach up at the bottom into the middle of the rib cage, you feel that little sticky outy point. Some people call it a solar plexus. Don't push on that too hard. And you see it's very little. It's kind of squishy. Yeah, but there's that part. So again, let's throw some skin on, take a little look from the surface anatomy. So again, the vertebrae, you can kind of push down here and feel where the spine is. You're going to look at a little bit of the curvature. So neck, right there is C7. We're going to go through the thoracic when it has that flexion part, again, roughly where the ribs are. That's a good way to landmark where your ribs are too, just purely from your spine. That little extension pattern right down at the bottom, that's your lumbar, okay? Front, we'll check out sternum. Sternum is going to line up right along here. You can feel that probably on yourselves. Xiphoid process right there, right kind of at the middle tent peak part of the rib cage there. Okay, let's scroll up a bit. Ilium. Okay, so now we're going down to the hips. Okay, so the ilium is going to be this bone here. Skeletor again. Again, sorry about the hands. I don't know why they loaded there. Tried getting rid of them a couple of times. They just kept coming back. And these lines shouldn't really be there either. Okay, that's Yeah, that's even a little more clear. Got rid of some connective tissue for you. Perfect. Always getting in the way, okay? So here we are. We have your pelvis, okay? And again, it's 3 bones. It's your sacrum back here, as well as the pelvic bones here on the left and the right. The ilium, that's this here. So if you look at the iliac crest, when you're talking about the ilium, you're really talking about this part right up here. So if you put your hands on your hips, right? So let's just bring back some skin right there. Hands on your hips, that's the part you feel stick out at the top. That's called your iliac crest, okay? I'll just stick there a little bit. That's a great view. I love that. Okay, so from here, we can also find the anterior superior iliac spine. And so if you look at the side view, I wish I could move the arms. If you look at the side view here, you know, and we're again just looking at the pelvis right there, that part that sticks out of the front of the hips. If I had this guy, I'd say right about there, I'd push in. But again, right there, anteriorly, this part where my mouse is, that's the ASIS, is how it's abbreviated. Anterior superior iliac spine, okay? Great. Flip around back, posterior superior iliac spine. So again, we know anterior, you know posterior now, they're just directional terms. Iliac, you know, superior, we know is above. Down here is inferior, superior, inferior. So this is just like a roadmap. It's very straightforward of what it's telling you where to go to look for this. Iliac spine, we know this is the iliac spine here. You put your hand on it. So again, anterior is front, superior is top. Iliac spine, so right up there. Flip it around, posterior, back here. What part? Superior, right. So superior and iliac spine, so it's right there. And the interesting part about this, and you may or may not have seen it, I don't know if it shows it on the model, not really, is back dimples, okay? So if you've seen back dimples kind of right back in the lower back, that actually coincide right here to some connective tissue to the PSIS, or the posterior superior iliac spine. And so that's going to come really handy in a lab coming up, okay? So make a note of this, memorize this one in particular. When we do a spinal flexion lab, you're going to have to landmark that. So being able to find the back dimples or the PSIS right there, which is harder when there's a bunch of skin and connective tissue over top of it, is going to become really important, okay? Moving forward, we're going to go down to the femur, okay? So again, let's take bony view. Great. Here's your femur, or the top of the leg bone or the thigh bone. Okay, it sticks into your pelvis there. And then you got this part that sticks out. That's called the greater tuberosity. So if we go back and do a half skin view, you're going to be able to feel that. That's the next bony, sticky outy part that's at the lateral side of your body there, okay? So again, that's your hip, and then this, the greater trochanter. Sometimes you just say GT. Your GT is the top of the femur there, sticks out of the side of the body, okay? If we keep moving down a little bit more, here's the condyles, medial and lateral condyles of the femur. So let's think back. Okay, condyle, that's a really weird word. You've already heard it once. Where was that? Right, back up at the humerus of your elbow, right? The medial and lateral epicondyles. Epi just means up or above. So this is the epi, the higher up condyles, inside and outside of the first limb segment from your shoulder, okay? And then very similarly down here, the first limb segment from your hips, you have the condyles, okay? Great. And how do you feel that when you got skin there? Well, it's just kind of the side of the knees, okay? You probably feel it stick out. You can kind of see it here. But as we flip around to the front of the leg, we're going to get to some fun stuff, all right? Oh, I need connective tissue for this one. Just wait and see. Check this out. Okay, so we got the patellas here, kneecaps, right? Same. Patella, kneecap, patella, kneecap. You know the patella bridge? If you are from the Lower Mainland, Metro Vancouver area, this is a great way to remember this. Let's throw on some connective tissue. Oh, that didn't help. Okay, let's try this. Oh yeah, that will. It's okay. All right, we need a little bit of muscle on here. Hopefully not too much. Okay, that'll suffice. Let's get in. We'll play around with layers as we get in. Let's dive into this knee. Check out the knees. We need to do this. Okay, so if we check out the knees here, look at the kneecaps, okay? Like, let's dial that all back again. There we are. So they're just floating out there. Look at that. Like, those are just bones floating at the knee. You know, you pass a deck of card through there or something, right? No, not quite. Why? Because those things are layered down or held in place by some tendons. Look at that. So your quad muscles attach to the patella and then back down to this lower bone, which we'll get to in a second, okay? The lower leg bone. So what's happening here? It's essentially creating a bridge to your patella, and they're called the patellar tendons, okay? Because they're really not creative when they're coming up with this stuff, all right? So again, patellar tendons attached to the patella. You can think of the bridge as bridging the kneecap across the knee, essentially, okay? Just like the patella bridge. All right, hopefully that helps you remember that a little bit. Some more connective tissue around the knee. Don't worry about that. Okay, let's get rid of those muscles. They're getting in the way here of our anatomy. So again, if we throw skin on, perfect. It's dressing up to head out, hey? So if we got that, we look at the knees. Here's our kneecap. Boom, there you are. You can feel that pretty readily. If you bend your knee back and forth, it'll move a bit. It should. Yeah, that's pretty easy one to landmark, okay? So now we'll move on to the tibia and the fibula. And this, again, I want to dial back. Oh, I want to get rid of some of the skin. Okay, so tibia and fibula. Okay, so you'll feel this just below the kneecap. That's the part that sticks out. And if you go laterally, you should be able to feel the head of the fibula, okay? And again, pretty common. So if you throw some skin on, let's try to landmark those. Okay. Well, this is probably what you ding on, you know, the side of your bed or whatever when you're cruising around the house. The coffee table is this, the tibia. It sticks out right at the front. And then you can't really see it. This is kind of a smooth over model, but the fibula I know is going to be right here, lateral to the tibial. It's called tibial tuberosity, right? Now a little sticky out part, okay? Great. All right, we flew through that. So that's the bony projections and landmarking those on the body. Hopefully that helps seeing with and without skin. I know sometimes it's a little hard and not quite intuitive if you're just landmarking on your body. I think that adds to a deeper understanding for you. And we'll just move on to the muscles now. So again, pause this lecture. Don't listen to me anymore. Take a little break. Go through this activity with yourself. You know, you'll need to look at the names and the words for the first while. But again, just try to go through it without any prompting. Rewind this. Go through it again. Try to landmark it as you go without looking. It should be intuitive where, you know, both for just carrying through your labs and doing exams and quizzes, you shouldn't really have to look at anything. You should just know where things are and be able to landmark them. It's going to make it way easier to communicate with your peers virtually, in the lab, anywhere when you're talking about this stuff, okay? So again, maybe a little pause, a little timeout, go back. We'll give you a little break to do that, and then come on back and we're going to do the muscles, okay? Great. Okay, welcome back. That was like no time for me. Hopefully you spent time going off and doing stuff. We're going to look at the muscles now. So with the muscles, I think I'm going to leave bones on. We'll start kind of with the upper trunk. Yeah, there we are. Muscle layer, for sure. Oh, that's not frightening at all. Okay, so we'll zoom in a little bit, just get a better look at things. Good. Yeah, I think that's the setup I want. Yeah. Okay, so starting off with the upper trunk. we're going to look at bicep brachii, okay? So biceps, a lot of you familiar with that one. That's the gun show, right? That's these here. And what's kind of interesting maybe is if we peel this back just a little bit, you get a better view. So you see those other muscles actually covered a bit. So when you flex your bicep, that's right here, okay? Yeah, great. All right. We'll throw back on some more meat. All right, triceps, that's the back of your arm. You can see that there. And some of these, you know, the triceps, you actually see a little V there. And the reason for that is you actually have 3 heads of the tricep. 1, 2, and 3. Okay, so you actually see that little V part differentiated. This is just connective tissue. You see the muscle in the back of the arm. Whereas the bicep has 2 heads of the bicep. You can see that kind of scooch up there, and that's why it's called a bicep. Okay, pec major. That's those right here. They go along the sternum and attach to the shoulder. Let's give you a little peek. Oh, perfect. Right there. You can see that attachment there is also where the next one, the pec minor, comes in. So this is your pec minor. Note 3 ribs up to the shoulders. Same insertion point here as the pecs pretty much. They have a couple of, they go to the humerus as well. But I think the big thing to note here is the pec minor, because this muscle is what's most common in what I call like the desk worker posture. So I'll just try to show it here. People's shoulder blades and shoulders protract. Remember protraction? So retraction, protraction. They protract because if you think of point A and point B here on this muscle shortening and tightening on yourself, turn it sideways. So if those 2 points get short, that pulls this whole shoulder complex forward, okay? And so once that goes forward, then these back muscles start to get long and tight, or loose, sorry. They get a little weaker, and that's where a lot of back pain for people come from. So it's actually this muscle up front causing pain and discomfort here in the back, okay? Lat dorsi. So lat, already we can think, oh, lat, where's that sound? Lateral. Let's go to the lateral side of the body. Whoosh, there we are. Great. And that's going to be this big honking muscle and connective tissue here, okay? Lats. A lot of people, let's throw some skin on again. Oh, a lot of people, you can kind of see that adds to that little bit of a V shape to a lot of people's bodies, where if these get quite large, you know, they kind of fan out quite a bit. They get too big, people can't even lower their arms, their humerus down to the side of their body, right? Because there's just so much muscle there underneath their arms, right? So that's the lats. You can kind of see it a little bit here. You know, it's the side of the body, but kind of there. Again, check that out on someone else. Everyone's bodies are kind of different. So being able to gauge and see where they are from a surface anatomy point is really useful. Okay, perfect. Let's get rid of that skin again. This one, we got a lot going on with this one. So with the trapezium, we have 3 sections of it really. We got the upper part here. Side view is a little better. Upper part here goes up to the neck. Okay, mid part is kind of from this upper section, goes down a little bit, and then your lower trap, oh, that's a better view. Lower trap here comes all the way down, okay? Mid, upper, mid, lower, okay? So you got 3 parts of it, all the same muscle. They all do different actions. That's why it's important to differentiate, although they all kind of work synergistically. For example, if I want to shrug my shoulder up, I'm going to use the upper trap. If I want to pull my shoulder blade down, so that's elevation and depression of the scapula, right? Which is this right here. You can actually feel that ridge of your shoulder blade or your scapula to be able to landmark the upper and lower parts of the trap, okay? Or the trapezius. Rhomboids, we got to peel back some of that skin and muscle again. So it's underneath. So how the heck do you feel that from there? Well, that's really tough, right? So we still got the lats here, but we got rid of the trapezium. So now we see these rhomboids here. Now, if you kind of think about it, again, if A to B shortens between these distances, not up and down, but left to right, what do you think that will do? This is your shoulder blade. So you take your shoulder blade, there's going to be protraction, right? Or retraction, sorry. It pulls everything back. It pulls this point closer to that point. That's what your rhomboids are for. And if the lower ones get a little more active, it actually helps pivot it, right? That's that rotation that we were gone over earlier. Okay. So again, through the skin, we're going to find where the shoulder blade is. So you'll feel the bottom of the shoulder blade, the ridge of the shoulder blade, and then between those 2 points running towards the spine, that's going to be the rhomboids, sorry, okay? Okay, skin off again. There we go, great. Throw on some muscle, boom, perfect. Wow, that's a very successful day at the gym, huh? Haha. It's a bad joke, I know. They're only going to get worse. This is what we're stuck with now, right? So with the deltoid here, that's the shoulder muscle, okay? So again, let's peel away, put on, again, right at the top, most superficial, right here. You can see that on our bodies without too much interference there. So let's go skin back again. There you go, top of the shoulders, perfect. Moving along, let's take a look at the stomach here and the abdomen. So when you're talking about the abdominals, you actually have 3 layers of abdominals, 2 of which we can only see. So the first one here, we look at the rectus abdominis. I'm going to try to get rid of any and all connective tissue. I think that's the best we can do. Yeah. So rectus abdominal, that's your 6-pack. You know, there's no way around it. This is the glory muscle when it comes to abdominals, you know. This and the subeguinal ligament, which isn't even part of your ab muscles, but it's the lowest ab kind of cut at the pelvis. But these give you the 6 to 8 packs, you know. Forward crunches do this. So again, if you think about A up here to B contracting, that's going to forward flex the spine, right? So if we're looking at that on the surface anatomy, pretty familiar, going to be right up in there, okay? Some people, again, depending on how much adipose tissue you have, can see them easier or not. Regardless of who you are and how much body fat you have, I promise you have a 6-pack under there. It's just whether you can visibly see it from surface anatomy or not, okay? But the next one might be a little more unfamiliar to people. We're going to look at the external obliques. So that's these running sideways here, okay? So that side view here is leading up to the 6-pack. That's your obliques, okay? And yeah, they cut off here, and yeah, they kind of lead to that cut down there on people that tend to be really lean. You can see the little curvature there, but really it's the inguinal ligament that pulls everything tight there. The obliques help a bit if you're really looking at that, but again, throwing some skin on is essentially at that border of the rectus abdominals and the side of the torso there, okay? The other thing people typically ask too is, what the heck are those things? You know, again, on some people you see them, they're not the lats, okay? You can see the lats here, and they kind of wrap around. If you look right in the armpit up high, that's the lat. But what the heck are those things? Are those ribs? Are those like chunks of muscle on the ribs to help you breathing? No, that's called your serratus anterior. And again, on some people you can see them. Let's see if our model has it. No. But normally they kind of pop out and they almost look like very bulgy ribs here at the side of the body. They're really not. They're just muscles. And their job is they connect to the shoulder blade at the back. They cause protraction of the shoulder blade. Okay, that's going to come up in one of your labs, but it's also just kind of fun to know. They're really kind of an odd set of muscles there for you to view. Moving along in your list, we have the erector spinae. So we're going to flip you around. And we got to muscle down a little bit here. Oh yeah. Can we do more? Perfect. Okay, so your erector spinae. So when we go skin on, you can see there's kind of like a bulgy ridge that run vertically on either side of your spine. You can probably even just, again, reach back and feel that. Kind of lean back a little, lean forward. It's like 2 tubes of muscle almost that run up and down either side of your spine. But what is that really? Well, peeling away that skin again, getting rid of that. Let's take a look. What it really is muscle that runs. That's the multifidus. It's kind of, okay. This will suffice. That's okay. You're actually feeling these bands of muscle that, in particular, these more medially towards your spine, okay? And you can see it's actually a series of muscles. It's not really just 1 or 2 single bands of muscle. It often feels that way, but they're actually tracks, okay? Yeah. Yeah, there you go. Erector spinae. That's even better. So this was the multitifidus, which are also involved in spinal extension, like the erector spinae. But we can even go down a little closer and see some lower spinal muscles too. It's hard to differentiate them at a surface anatomy level. You don't need to worry about that. Okay. Yeah. Okay. Not a great view, but that's okay. You can definitely feel it from a surface anatomy perspective, okay, and see it again, right along there. Okay. Moving on to the legs. Perfect. Here we are. So quadriceps. That means 4. And surprising, peeling back, what do we see? Well, we can only really see 3. So 1, 2, 3 muscles on the upper thigh. I think we have to go a little deeper. And yeah, it's not really showing it. It is underneath there. There's a 4th one called the rectus femoris. No, it peels back too quick. But anyway, so you got 3 of the 4 quad muscles up here that are readily palpable, or you can feel them, okay, from surface anatomy. I'm going to flip this person around. Then we got hamstrings. And you see there's a whole bunch of tubes of meat, or muscle, if you will, here. But really, you're talking about these 3 muscles, the hamstrings. Interesting thing about the hamstrings, and again, I hope we can see this, is it goes over the knee and hip. Yeah, so here comes the hamstrings attaching to the pelvis, so it helps move the pelvis. But also, if you look here at the knee, I'll zoom in a bit. You can see it actually articulates over and across the knee here. Okay, oh, that's a bit messy for you. Give me a sec. Yeah, so you can see the hamstrings here, these 3, come over top of the knee, which means it articulates at the knee too. Most muscles only work across one joint, you know, aside from the spine. So when you're talking about our limbs, the hamstring's a little different that way. Okay, great. Good. Gastrocnemius. Yeah, got to keep heading down. So we have 2 different parts of the calf muscle. Okay, so that was the quads again. You can see them. Sorry, just to review that. Hamstrings, you can see them here. Some people have quite a predominant hamstring across the knee. That'd be the easiest to see. But it's the front and the back of the thigh, or the femur. Okay. Moving down here, calf. Okay. So we have the gastroc, or gastrocnemius, which is the part that sticks out. And again, you can see a little bit of a bulge here, typically on most people. That's like the power producer. So this is where a lot of force comes in your ankle flexion and extension. Sorry. And then you have a soleus with that's underneath it. So let's take a little peek, diving in there. Okay, zoom. All right. So you have the gastroc up here, gastrocnemius, and this soleus that comes down over top of the heel. This is actually your Achilles tendon. You see that there, kind of sticks out. That you can feel too. It'll be like right above your ankle, just a tight little band. It's really, really strong. It has to deal with a lot of force production from your lower body. But this part is the soleus, and the soleus actually creeps up underneath here. You can kind of see that in this diagram. And the gastroc are these 2 big protruding muscles. Again, kind of gives that little dimple, almost tricep looking effect down on the back of the calf there. Okay. Last but not least, we'll scooch back up to the glutes. Okay, glute is another way of saying your butt muscles, but there's actually a few of them. So there's the glute max. We'll peel this back so we can take a look. The glute max is this here. Okay, but a really, really crucial and important muscle is actually your glute med and glute min. Okay, so that's these over here, and they are super important for balance on one leg. They're also super important for low backs, as well as just general posture and stability control. It's all in the hips. You know, if you can think back, Shakira, the hips don't lie. It's all in the hips. There's been studies, for example, done out of the University of Calgary and repeated elsewhere, that 95% of knee pain occurs because of a weak hip, and in particular, these muscles. The glute max is definitely involved, Again, when we extend this research onto, say, running and lower body injuries, so if you're a soccer player in particular, this is really, really crucial. A lot of your lower body stuff is coming from a weak hip coordination, typically, again, 90-95% of the time, okay? And most of you, it'll be hard to feel, but you remember this thing? What was this? That's your, iliac crest, right? You can feel that. ASIS, PSIS. This is your greater trochanter, right? That's your femur. So if you palpate that and palpate your ischial crest, you're gonna know roughly where the glute med and min are. Again, more posteriorly, and for good reason. For your posterior is the glute max, okay? And that's more for leg extension, okay? So hopefully putting that into context a little bit was helpful as far as knowing what these muscles are kind of involved in. The degree that you're going to need to know them is kind of where to find them, how to use them. You're going to have labs that are going to direct you to landmark your PSIS, landmark the lateral epicondyle of the elbow, you know, and so being able to be familiar with these, or knowing that, say, your quad muscles help extend your leg, and using that terminology, extend, flex, is going to be really helpful, again, both virtually and continuing on as far as describing and communicating with anyone in the health profession, or with your peers during these labs. Okay, so be sure to go back and review everything at least once or twice. Again, you should be able to landmark a lot of these things moderately comfortable. The more you use this language, or try to, when you're doing your labs actively with your peers, or communicating in reports or discussion with myself, I'll be looking for that. To use this language, so you're going to practice it. It's going to become second nature. If you're not sure, it's okay. Take a guess. Say the wrong term. One of your peers or myself are going to know, and hopefully we'll be able to help you out with that. Okay. So in the end, being specific and accurate with your terminology that you use, again, with myself, with your peers, with healthcare professionals down the road, whatever it happens to be, just expedites communication so much. You know, you can think of a bunch of surgeons sitting around an open body and being like, oh, move the thing on the left. What thing? Your left, my left, you know, up or down. Well, is this up and down, or is it up and down on the body? You know, it can get really confusing really quick. So again, being familiar with all of these terms just expedites your understanding and communication with everything to do with the human body as you move forward. So with that, have fun trying these things out on your own, landmarking things on people that are around you. Anyone willing to participate and be a volunteer for a bit for this, I highly suggest you use them. And yeah, play around with it. Again, it's okay to get some of these terms wrong when you're trying to work with peers, but it's better to try using them and then learn and move forward, and then you don't have to come back and study this stuff. It's just stuff you kind of know. Okay? All right. Best of luck with that, and look forward to seeing your discussions with your lab activities for this section. Take care.